In transduction, DNA from the chromosome of one cell is transferred to another cell via a replicating virus. A virus that infects bacteria is called a bacteriophage or phage. There are two types of transduction, generalized transduction and specialized transduction. Generalized transduction, as shown here, is initiated during the lytic cycle of virulent bacteriophage. The donor DNA can be any fragment of the bacterial chromosome. Specialized transduction, which is shown on a separate animation, is initiated during the lysogenic cycle of a temperate bacteriophage, and the donor DNA is a very specific part of the bacterial chromosome adjacent to the prophage. At the beginning of generalized transduction in bacteria, a bacteriophage attaches to a bacterial cell wall and inserts its DNA. The phage commandeers the bacterial cellular machinery to synthesize the new phage DNA and produce phage proteins. In order to completely convert the bacterium into an efficient virus-producing factory, some of the phage proteins break the bacterial DNA up into small pieces. Normally, phage DNA is coated with phage protein to form new virus particles, but sometimes pieces of bacterial DNA are mistakenly packaged in phage proteins. These sloppily packaged phage particles carry bacterial DNA rather than phage DNA and are called transducing phages. When a transducing phage, composed of phage proteins and bacterial DNA, infects a new host, the phage injects the bacterial DNA into the new bacterial host. The infected bacterium sometimes incorporates this DNA into its own chromosome through recombination, resulting in the transfer of genes from the donor bacterium to the recipient bacterium.